Great. No, computer is freezing up. Um, just a moment. I'm having some issues with computer freezing up. Okay, seems to be working now. So I'm going to go over the last of the chromatography stuff today, and then on Tuesday next week, because we won't meet on Thursday this week, we'll start NMR. Let's go over the last of the chromatography stuff. So we'll do some last bits. So supercritical fluid chromatography is a sort of hybrid, is a literal hybrid of gas and liquid chromatography that does combine some of the best features of both. So supercritical fluids exist at temperatures and pressures above their critical temperature and pressure, and will have densities, viscosities, and properties that are sort of intermediate between gas and liquid. So when you take a substance above its critical pressure and temperature, there's a range, and you get this state called a supercritical fluid, which is sort of in between gas and liquid. So for example, I have a gas. For gases, the densities are between about 0.6 and 2 times 10 to the minus third. For liquid, they're between 0.6 and 2, and the supercritical fluids are in between around 0.2 to 0.5. Diffusion coefficients are in between, viscosities are in between. And the critical temperature is the temperature above which a distinct liquid phase does not exist, regardless of pressure. So here you can see for a pure substance, there'll be a solid region, a liquid, a gas, and then up there's a point, the critical point, sort of above which you have the supercritical fluid region. And some of the properties that are nice about supercritical fluids is that they're remarkably able to dissolve large non-volatile molecules. So supercritical CO2 can dissolve N alkynes containing over 30 carbon atoms. This is related to their high densities. The dissolved analytes are easily recovered because you can equilibrate with the atmosphere at relatively low temperatures, and the analyte and supercritical CO2 can be recovered by basically reducing the pressure and allowing the CO2 to evaporate, turn into a gas. when you don't need organic solvents, so they're environmentally friendly. They're inexpensive, innocuous, and non-toxic. They're primarily doing this with CO2. And they have higher diffusion coefficients and lower viscosities relative to liquids, which allows for faster, higher resolution separations. And much for these same reasons is actually why so you can use them for other things like dry cleaning. They use supercritical CO2 for dry cleaning in some places. So supercritical fluid chromatography can separate compounds that aren't conveniently handled by gas chromatography or liquid chromatography. Things that are non-volatile or volatile or thermally liable, and contain a functional group that makes detection in liquid chromatography using spectroscopic or electrochemical techniques easy. So about 25% of separation problems fall in this category. So things like polymers, fossil fuels, pesticides, foods, drugs. The separations are faster than liquid chromatography. They run at lower temperatures than gas chromatography. And this is beneficial in industrial scale purification. So they're faster than liquid chromatography, lower temperature than gas chromatography. So here's a comparison between high performance liquid chromatography and supercritical fluid chromatography. And the instrumentation for supercritical fluid chromatography is very similar to ordinary high performance liquid chromatography equipment, since the temperature and pressure requirements for supercritical fluids fall within the same range. The two major differences is that you have a thermostated column oven, because you require precise temperature control of the mobile phase, which is typically supercritical CO2. And you have to have a resistor or some sort of back pressure device that's required to maintain the desired pressure in the column, because it needs to be at high pressure. And you want to have a change in pressure to convert from supercritical fluid to gas to transfer it to the detector, because detection is much easier when it's a gas. But it's fairly similar to high performance liquid chromatography. And pressure has an effect where an increase in pressure results in reduced dilution time, because this increases the density of the mobile phase, which affects the retention or capacity factor. And pressure changes are analogous to the gradient dilution in liquid chromatography and gas chromatography. So what properties of a supercritical fluid are important in chromatography are the fact that it can dissolve large molecules while still kind of acting like a gas, and that it is working in a lower temperature regime. Another chromatography-like studying that you probably have some exposure to is electrophoresis. It's a technique where solutes are separated by their different rates of travel through an electric field. And this is most commonly used in biological analysis, particularly in the separations of proteins, peptides, and nucleic acids. Those DNA divisions. It's similar to chromatography. Both involve separation by different rates of travel through the system, but in electrophoresis, the different rates of travel are produced by an electric field. So you've probably seen gel electrophoresis. And in chromatography, the different rates of travel are due to chemical interactions between the solutes in a stationary or mobile phase. So the theory is that the migration rates of solutes in an electric field will depend on the size and charge of the solute, as well as some other factors. And the degree of retention or rate of travel of a solute through an electrophoretic system is given by mu times the electric field strength, where mu is the electrophoretic mobility of the solute. And it's a constant for a given solute under a particular set of experimental conditions, depends on the ionic charge of the analyte, and it's inversely proportional to things like the size of the analyte, the shape of the analyte, and the viscosity of the medium. These are the things that slow things down. So a charge to size ratio of an analyte is an important factor in its mobility. Small or high charged molecules move faster, large or low charged molecules move slower. So it can be performed in an open tubular system where you're driving it through a fused silica capillary, and it comes out over time. So you're detecting how it comes out over time. Or a packed bed system where you're running it generally through a gel. And it's, electrophoretic gels are commonly done. This is more common in biological analysis. You have several different sample wells and you look at the progression through the gel. So some issues that can happen are Joule heating, which is uneven heating in the system caused by the electric field, which causes different points in the system to have different temperatures, and you get a non-uniform mixing of the solute and solvent. This causes peak broadening. To decrease the band broadening caused by Joule heating, you can use packed bed systems or stabilizers that prevent the mixing of solvent from different regions, and use more efficient cooling that prevents the formation of thermal gradients. So this is an example of Joule heating occurring. Zone electrophoresis involves applying very narrow bands of sample to the electrophoretic system. A very common type of electrophoresis. You have a constant buffer composition throughout the system, and the solutes can be totally separated in a reasonable amount of time into separate zones based on their mobility. And the zones are separated by regions of buffer. And then, so this is the example, you load samples into little wells in the gel, and then you visualize the bands of different samples with UV light, for example. So you may have done this in biology class. Packed bed or slab. So much of the work in zone electrophoresis uses packed bed instead of open tubular supports. This avoids broad banding from Joule heating and unstable density gradients. Packed beds stabilize the solvent in the system. It prevents mixing between different regions. So some common supports are cellulose acetate, paper, agarose, and polyacromide gels. Band broadening can be caused by diffusion across particles around particles in the packed bed. And a porous matrix may also result in molecules being separated according to size in addition to electrophoretic mobility. So here are some examples with DNA samples or blood stains being separated by electrophoresis. Capillary electrophoresis is another type of zone electrophoresis. It uses a narrow, open tubular supports or capillaries, typically 25 to 75 micrometers inner diameter. The narrow bore capillaries allow for the efficient removal of Joule heat from the system, which decreases band broadening. This allows stronger electric fields to be used, which leads to faster migration of solutes and decreasing the analysis side. Capillary electrophoresis is nice because it's easier to automate. You can use very small sample sizes like nanoliters. Because there are no particles, there's no multiple paths. And many types of detectors can be used in capillary electrophoresis. Fluorescence or UV absorbance is commonly used. So something like this where you have a photocathode, your light source and a photodetector. And because of the extreme efficiency and narrow peaks, it's an area of intense research, especially in areas of biotechnology and biochemical separations. It also has the nice advantage of very small sample requirements, so it can be useful in separating and studying the ionic solutes present in single cells. And it can also be useful when screening large chemical libraries for drug discovery, or genome sequencing is the other use it can be useful for. And all solutes, regardless of charge, will migrate toward the negative electrode of the system because of electroosmotic flow. This is a feature of capillary electrophoresis. It's produced by the presence of negative charge on the surface of the capillary. The negative charge is produced by ionization of sinyl groups on the capillary surface at pHs of 4 to 5. And a net positive charge needs to exist in the solution to counter the negative charge on the capillary surface. And the net positive charge of the solution causes net migration towards the negative electrode. So all solutes will eventually pass the detector regardless of charge, which is useful, instead of only detecting, say, positively charged solutes. But more positively charged things will move faster, where negatively charged things will move slower. So if you just have electrophoretic, positive would go one way, negative would go other way. But if you add the electroosmotic flow, you get everything going towards the cathode, but the more positive goes faster. The flow profile here is flat. There's no peak broadening. The pressure-driven flow in liquid chromatography will cause a parabolic profile by boundary effects and inertia, but electroosmotic flow does not have that effect. The electroosmotic flow looks like this, while hydrodynamic flow looks like this because of the interactions with the wall. And then pH can affect charge, which can affect the rate of migration. And then also for a protein with amino acids, they can switch from at a low pH, they might be acidic. At a high pH, they can change their ionization. And the isoelectric point is the pH where the net charge on the molecule is 0. So, and if a plate has a pH gradient, proteins will migrate until the local pH is at the isoelectric point. So free large proteins are ionized at the pH where the capillary electrophoresis separation is carried out. If they're A, 2+, B+, and C3+, you can predict the order of elution. Well, I would expect C to come first, then A, then B, because the larger charge is going to move faster. And that was all I really wanted to cover for today. That was short, but we didn't have that much to go over. Any questions? We will start up on NMR next week. And we will not be here on Thursday. Not, well, then I'll let you guys go. And have a nice day. There's some questions. Okay. Okay. Have a nice day.